Hello. Today I'm going to talk about COVID-19 vaccines, which have been developing at a pandemic pace. And I'm going to focus my talk today on the latest results from clinical trials. I don't have any conflicts of interest, personal conflicts of interest to discuss. So I'm going to start by talking about natural immune responses to SARS-CoV-2. Because all vaccine development starts with understanding what our natural immunity is. So in this particular study, 149 individuals who had recovered from COVID-19 were looked at. And what these investigators found was that there was a wide range in the immune response that developed in these individuals, with in particular a wide range in their neutralizing antibody responses. These responses are shown here in this figure on the right, which stratified them by those that patients that remained outpatient and those that had been hospitalized. So here they found that about a third of people who recovered from COVID-19 had pretty low titers, whereas there were also a couple of individuals that had extremely high titers. So in general, it's a wide range. These investigators also did a deep dive into those antibodies to see what they really looked like. And what they found is that the antibodies from different individuals were actually quite similar to each other. They all targeted the spike protein, which is the protein on the surface of the virus. And they also targeted the same actual epitopes of that spike protein. In addition, even if they found them at only low levels, they did find potent neutralizing antibodies in all of the individuals that they looked at. This is really important for vaccine development because it supports the concept that a vaccine can work in a broad spectrum of individuals. It means that all of us can potentially make a neutralizing antibody response, which we think is critical for protection. There's some additional data emerging about the durability of these immune responses. This is a study that was done in Iceland, was published this month. They looked at over 1,000 individuals who had COVID-19. And they looked at them looking at these antibody responses over time, and that's shown here in this figure. Again, they stratified those participants by those that had been hospitalized and then over the time that they had recovered. So they found that 91% of the patients they looked at were seropositive, meaning they developed antibodies by 25 days, and that those antibody titers increased over the 2-month period and then stayed steady. What they found at 4 months of follow-up is that there was no significant decrease in those antibody titers. Hospitalized persons seroconverted more frequently and quickly, and antibody titers were most strongly associated with hospitalization and clinical severity. But what's most important for us in terms of vaccines is that this data provides evidence that antibodies can persist at least over a 4-month period. We don't know too much yet about whether antibody responses in humans actually protect us from reinfection, but we have looked at this question a little bit in non-human primates. So this is a study that came from the group where I'm based, the Center for Virology and Vaccine Research in Boston. So what my colleagues did in this experiment is they had 9 rhesus macaques, and they were asking the question if animals can get reinfected with COVID-19, meaning if they got infected once and then recovered, could they be infected again? So what they found first is that when they challenged these monkeys, which is shown here, that they, the first time, they all had high viral load in their lungs. And this peaked in the first few days and then came down quickly over a week and then fully resolved by 3 weeks. These animals, after this challenge, all recovered and developed antibodies, neutralizing antibodies. At day 35, so not too much long afterwards, the animals were re-challenged again. But this time, this is shown in the figure here, was a very different response. The second time, animals, only 3 of them had low viral load in the lungs with no recoverable live virus at all. And no viral DNA was detected in the lungs at any other time point past the first day. And these animals all had a rapid boost in their immune response, showing immune memory. So this data is really showing that natural immunity is possible, and at least it was demonstrated in monkeys. Our group has also been able to show, along with many others at this point, that vaccines can induce this immunity, and it's also protective, at least in animal models. So in this experiment, my colleagues used an adenovirus 26-based vaccine. That's a viral vector that expresses the full-length spike protein. They actually immunized 52 monkeys with some different variations of the spike protein. And then again, they had some that got placebo. These animals all developed neutralizing antibody titers, and the best vaccine was the one that expressed the full-length spike protein. This particular vaccine that I'm showing here is the one that's progressing in clinical development. The antibody titers in these animals were quite high. They were actually 4-fold higher than what was observed in convalescent humans. So it's possible we might be able to do better than what's seen in humans with vaccines. And these monkeys had nearly complete protection after they were challenged. So here's the animals that got the placebo. They all had virus in their lungs. And here are the animals that got vaccinated with the best vaccine, and they did not have virus in their lungs. In addition, it looked like neutralizing antibody titers inversely correlate with protection. And this is also shown here where we separated out the animals by those that were completely protected versus partially protected for animals that got the different vaccines that weren't as effective. And this is showing that neutralizing antibodies are a correlate of protection and may be a mechanism of protection. A follow-up study also showed that these vaccines can protect animals against severe disease as well. So now I'm going to move on to the clinical data. So there are a lot of different vaccines that are in clinical development, and it would take days to go through them all. But what is important to know is that those that are in advanced clinical testing all share the same spike protein. They all express spike protein. It's just different mechanisms of delivery. Today, I'm going to focus on 6 vaccines. These are ones that are in phase 3 efficacy testing, but that also have published clinical data. And here are the 6 vaccines I'm going to review today. For this talk, I'm going to refer to them by the pharmaceutical companies that are sponsoring these trials and producing these vaccines, though most of these vaccines are developed in collaboration with academic groups and also government groups. If anyone is interested in doing a deeper dive or just checking in on their favorite vaccine, I recommend going to the New York Times, which has a coronavirus vaccine tracker, and that will tell you about the entire pipeline of vaccines. So I'm going to start by talking about the Pfizer vaccine, which may be the most developed or advanced in development, at least in Europe and the United States. So this vaccine is a lipid nanoparticle that encapsulates a modified RNA that expresses the spike protein. Now they haven't published this data in a peer-reviewed journal, but they did make it available in medRxiv, and they put up their phase 1 data. So what they did in this phase 1 study is they did a randomized controlled trial, and they had about 90 people who received or were in the arm that got this BNT 162b2 vaccine. The study was actually much larger than that. It looked at additional vaccine, but today I'm just going to tell you about this one because this is the one in phase 3 testing. So they looked at 2 vaccinations 21 days apart, and they looked at 3 different doses. And they also looked in young people, 18 to 55, and then an older group, 65 to 85. So what I'm going to do for this vaccine and all the others is I'm just going to tell you first about what was reported for vaccine safety and then what was reported for the immune response. So here the vaccine safety data show that there were no serious adverse events and no study pauses, but strong big caveat that they reported only 7 days of follow-up so far from their second vaccine, so not a lot of time. The symptoms that they observed were relatively mild. You can see here that the majority of participants actually had no symptoms at all, and really fevers were very uncommon following immunization. Symptoms were somewhat dose dependent. Here's the 30 microgram dose, and they were higher after the second dose. They actually went up with the second dose, but in general, it was very well tolerated. The antibody responses for this vaccine were quite good. So anti-spike antibodies were generated in all of the participants after the first dose. But for really good immune response and neutralizing activity, it really required 2 doses. So in this figure, you can see these small bars are showing the responses after the first dose, and then these big bars are after this boost. And then the immune responses were seeing the same pattern in both the younger adults and the older adults. The lowest response was with the 10 microgram group. And importantly, even in the older adults, the titers were as high as convalescent plasma, which is shown here, convalescent serum, and actually were higher, significantly higher for the younger adults. And they didn't have any data to share yet for their T-cell responses. So the sister study to Pfizer, or sister platform, is the Moderna vaccine. And this is also a nanoparticle that also encapsulates an mRNA. So very similar concepts. Here they've published their phase 1 data in the New England Journal. This was done a couple of months ago. Now the study design for their phase 1 was an open label trial with 45 participants, 2 vaccinations, 28 days apart, with 3 different doses. These are some higher doses than what was tested in Pfizer. So here they found no serious adverse events and no study pauses. And after the first dose, symptoms were relatively mild, and probably a majority of participants didn't even have systemic symptoms. But after the second dose, it was quite interesting. There was a big jump in their systemic symptoms. You can see here that 100% of participants at the higher doses had systemic symptoms. And actually 21% of those at the highest dose had severe symptoms, which in a clinical trial means that it really interfered significantly with their daily life, and required, in a lot of cases, required some sort of medical treatment or intervention. These severe symptoms were essentially a febrile syndrome, so fever, malaise, chills. And here, about half the participants had fever, sometimes as high as 103. Their immune data was great too, similar to Pfizer. So anti-spike antibodies were generated in all of the participants. And neutralizing antibodies, again, required 2 doses. This is for the high dose. You can see that there's a little bump in immune responses, but really after the boost, a big bump. The lowest responses were with that low dose. And what's really cool in terms of their immune responses is that they were really at the higher end of what's seen with convalescent serum. T-cell responses were characterized in this study. CD4 T-cell responses were detected, and they were strongly biased towards what we would call a Th1 phenotype. And this is relevant because we are looking at the risk for vaccine enhancement of disease, which is typically associated with a Th2 biased result. So this is supportive that these would be safe. So moving on from the 2 RNA vaccines, I'm then going to discuss the Astrazeneca vaccine. And this is a different platform altogether. This is a viral vector vaccine. It's based on a chimp adenovirus vector, again, expressing spike protein. This paper came out in Lancet. It actually summarizes about 1,000 participants' data, so quite a bigger study. And here it was an interesting design. They looked at 5 × 10⁻⁻⁻ viral particles, basically 1 shot of this, and almost all the participants, it was just 1 dose. And they didn't compare it to saline. They compared it to a meningitis vaccine, which has its own little profile of reactogenicity. They also had 1 arm of the participants that received prophylactic paracetamol or antipyretic, meaning before they got the vaccine, they took that dose. In terms of vaccine safety, there were no serious adverse events and no study pauses in this phase 1/2 study. This was a much more reactogenic or symptomatic vaccine than what I reported earlier, at least in terms of after the first dose. So after just 1 dose, systemic symptoms were quite common. You can see them here. It's this febrile syndrome, fever, chills, joint pain, malaise. About 60% of participants experienced that, and there was quite some high fevers. So 2% of participants had temperatures over 102. Yes, taking antipyretics did prevent many of those symptoms, but it's unknown fully whether that would affect immunogenicity. Here, in contrast to the RNA-based vaccines, symptoms after the second dose were actually less severe. So they had 10 participants that they boosted, so not a lot, but in that small number of people, the symptoms seemed to go down the second time. The immune data, interestingly, was, to my eye, not quite as good as what was seen with the mRNA vaccines. Here, anti-spike antibodies peaked at about a month. Now, remember, most participants only got one shot. And neutralizing antibody titers were relatively low after that single dose. So granted, I'm comparing kind of apples and oranges because we're looking at one dose versus two, but the one dose results were really at the bottom of what would be seen in convalescent plasma. In the 10 individuals that got boosted, those responses did go up, which is great, and they were more comparable to convalescent samples, though not necessarily at the high end. So I think this is showing that for this vaccine, I think a single dose is not going to be enough. T-cell responses were detected, but they didn't report on Th1 or Th2 phenotypes. So now I'm going to talk about Novavax, and technically this phase 3 study hasn't started yet, though it's supposed to start in a couple of weeks, but they did publish their phase 1 data in the New England Journal, and for that reason I wanted to share it with you. So this is also a nanoparticle-based vaccine. So this is similar technology to the mRNAs, but instead of encapsulating the RNA sequence, it encapsulates the spike protein itself. And this was administered with or without an adjuvant. This was a randomized placebo-controlled trial, which they conducted in Australia. They looked at 2 doses. They looked at 1 or 2 administrations of that, and then again, with or without this adjuvant. In terms of vaccine safety, this was a pretty well-tolerated vaccine. There were no serious adverse events, no study pauses, and the majority of individuals, I mean, look at after the first dose, really the majority had no symptoms, or it was very similar to placebo. Only 1 participant in the whole trial after any of the doses had a fever. There was maybe slightly more symptoms after the second, but still very well tolerated. And the antibody responses were also quite good. So a single shot did elicit binding antibodies in nearly everyone, but the boost really did make those antibodies jump. The adjuvanted 2-dose regimens were the most immunogenic, 2 doses with adjuvant, and that it really didn't make a difference at the highest doses, whether or not they both performed similarly. So here is 5 micrograms adjuvanted, 25 micrograms adjuvanted. You can see how the boost really raises up those values. And these high values that were observed were actually 4 to 6 times greater than matching convalescent serum. This is a contrast, I think, to the chimp adenovirus vaccine. Granted, if you recall, that was only 1 dose. Okay, so I'm going to end by talking about 2 vaccine programs that are outside of the US and Europe. And the first is the Ad5 vector vaccine program in China, which is the CanSino vaccine. This is a replication defective Ad5 vector. They were actually the very first to publish human data. And in fact, they're the only ones that I can see that have 2 separate peer-reviewed publications for their phase 1 and their phase 2 data. So they're really on top of it. Their phase 1 trial was an open label study in Wuhan. Phase 2 was randomized controlled in Wuhan. In the first study, they found, they tested 3 doses, and they found that the high dose was really not well tolerated. This kind of febrile syndrome was seen, and the neutralizing antibody titers after a single shot were not fantastic. They were low and moderate. The phase 2 study, it was interesting. They dropped the high dose and went with just low and middle, but here they found that even the middle dose was not well tolerated. Actually, 9% of the participants had severe adverse events, mostly with those temperatures of higher than 101.3. So it's in Celsius, getting around 39 in Celsius. And the low dose is immunogenic as the high dose. That's seen here. But to my eye, these neutralizing antibody titers are not terribly high, and they don't compare them to convalescent sera in this paper, but I think they're probably lower than observed with the other vaccines. But again, 1 shot. So if it's 1 shot, you know, who knows, maybe a second shot would boost it. Phase 3 trials are underway in China and the Middle East, but I mentioned here that there's actually limited approval for this vaccine in China, where they recommended it or are administering it to Chinese military, but it's unclear the details of that. The last vaccine constructs I want to talk about is the controversial Russian vaccines. Now, what they've reported is that this was an Ad26 prime followed by an Ad5 boost expressing spike proteins. They did publish some data in Lancet just a couple weeks ago, which they call a phase 1/2 study, though I have to say that the sample sizes are quite small to be really considered phase 2. What they looked at, both of these are open label trials, and the vaccine combination, which is what's in going into clinical use, they really only reported data on a total of 40 participants between the two studies. What they reported is that, consistent with prior studies of these vectors, they were generally well tolerated. About 50% did have fevers, so this seems to be common with these vector vaccines. And then the neutralizing antibody titers look to be, here you can see them here, they look to be comparable to convalescent plasma. So not those 4 times as high and not low, but like right in the middle. So the phase 3 study is apparently underway, but there's not a lot of details. What makes it very controversial is that it was supposedly approved in Russia and recommended for use, but so far it's actually unclear what the uptake for this is. And I just want to kind of point out again that this is all very, very small sample size for a safety cohort. So there's really not a lot we can learn from this, even from this published paper. So now I'm going to summarize what I've told you today. So COVID-19 leads to neutralizing antibody responses in humans. There's a wide range in the titers. It's likely related to the severity of illness. Neutralizing antibodies target the spike protein, but it's unknown at this moment, the data is developing, if these neutralizing antibody titers are actually protective in humans. Animal studies suggest that immunity to COVID-19 is possible. This is shown with convalescent neutralizing antibodies that were protective against reinfection, and also that vaccines can protect animals from COVID-19 infection and severe disease. Nearly all of the COVID vaccines that are in development use the spike protein. It's just the delivery mechanism that is different. Finally, phase 1 and 2 clinical data really are promising. Multiple platforms were shown to be safe and generally well tolerated, depending on the doses. The vaccines all elicited antibody responses of one kind or another. There's definitely room for improvement. Some participants had a fairly severe but transient febrile syndrome that was kind of dose limiting in a couple of these studies. And also really all of them showed that 2 doses appear to be required, at least with these platforms, for a strong neutralizing antibody response. So the single shot for these vaccines didn't seem to do enough. And then finally, I want to remind everyone that the durability and the protective efficacy is really currently unknown for all of the COVID-19 vaccines at this time. So none, I have not seen any data yet that showed for sure that these vaccines are effective at preventing COVID-19. But hopefully we will see that data in the months to come. Thank you.